So today we discussed about the plant tissue culture. That man is dependent on plants for food, shelter, clothing, medicine, air purifications, and any other things. It is the dependence of man on the plants that has led him to discover different ways to protect and preserve them from beginning loss of natural and man-made calamities. While scientists hit upon the technique whereby plants can not only be protected from being lost but are able to develop into a complete plant from a single cell or a small plant part. This technique is called tissue culture, subsequently proved to be a boon for the mankind. So, from a single cell or small part of the plant, if grown in a complete plant, from a single cell or a small plant part, the whole plant be developed, that is called the tissue culture. That is due to the ability of tissue culture. So, getting a complete plant, that is, getting a complete plant from a single cell or small plant part, that is called plant tissue culture. Theoretically, any plant cell except those lacking of a nucleus enclosed by rigid and secondary wall is potentially capable of regenerating the organism, that is plant. Any plant cell except those lacking of a nucleus or enclosed by a rigid or secondary wall is potentially capable of regenerating the organism, that is plant, from which it was taken. Such a cell is said to be totipotent. I mean, a cell which possesses nucleus or not by enclosed by a rigid secondary cell wall is potentially capable of regenerating the whole plant from which it was taken. Such cell is said to be totipotent. Collection of similar cells from tissues and tissue systems are organized into organs, and specific spatial arrangement of organs constitute the organism from a organ plants, or it develops into shoot tips, root tips, leaf, primordia, developing embryos, bud cells, and so on. Tissue explants cells, the parenchyma, collenchyma, and protoplast. That so a cell, you know that some cells of plants that have no nucleus, that is means in phloem, companion cells. You know that two types of vascular tissue, xylem and phloem, and phloem has a mixture of four types of cells, with a companion cells, sheath tubes, phloem parenchyma, phloem fibers. So in companion cells of phloem, there is no nucleus. So except the companion cells, any other cells which not possess the rigid cell wall are capable of regenerating the whole plant body, that is called totipotent. And from that totipotent development of a whole plant is called your totipotency. That's called your totipotency. The ability, the ability or capacity of mature living cells from the plant body develop into so now getting a plant from totipotent is called totipotency. The ability or capacity of mature living cells when free from plant body develops into new organism in controlled condition is known as totipotency. So that single cell which is totipotent that develops into a new plant that should be under control condition. Under control condition, that totipotent develop a new plant, develop a new tiny plants which is taken from it. That is called totipotency. Every living cell of the plant body able to regenerate the whole plant body. Every living cell of the plant body able to regenerate the whole plant body because it derived from the fertilized egg and contain hereditary information. Hereditary information. So every living cell of plant body have the capacity to regenerate the total plant body as because it is developed from that fertilized egg and contain hereditary information. And this called your cellular totipotency. Cellular totipotency is a property of a plant cell because differentiated plant cells remain in capacity to give rise a whole plant. But in animal cell, it loses the capacity of regeneration after differentiation. Whereas totipotency is more easier or maximum possible in case of plant, but in case of animal that is not, as because animal cell loses the capacity of regeneration. Only few cells of animal cells, that is bone cells, possess the power of regeneration some extent, not completely. Some extent possess the power of regeneration for which breaking bones are joined again. That is due to the regeneration. But old parts has not the capacity for regeneration. If a plant body cutting from the top or any plant body cutting from the some extent from base of the height, then that germinate, again budding develops and that buds again develop into new plants. But if animal, if cutting from head or any part that did not never regenerate or regeneration capacity, animal cell not possessing maximum, only few, some extent that muscle cells and bone cells possess some extent for regeneration, which is not so maximum. So totipotency is not never found in the animal cells because regeneration capacity is very minimum. Nerve cell or neuron cell not possess the capacity for regeneration, as in case of animal. So Stewart, a slice of phloem tissue of carrot grown a liquid nutrition medium containing coconut water. The part of the plant is taken for tissue culture is known as explants. The part of the plant is taken for tissue culture is known as explants. Example, phloem tissue of carrot root. The liquid nutrition medium containing explants was allowed some hours for shaking. Then the culture cell divide continuously to form mass of undifferentiated tissue known as callus. Some of these cells clusters started differentiating the initial of root. When it transferred to semi-solid medium, then it develops shoot system and gives rise to new plant. The plants then transfer to plot or soil where they develop in a new plant. So, if you take some phloem tissue of carrot from the root, then it taken in a liquid medium of coconut, then that develop a new plant part. In plants, even mature can be de-differentiated, divide, come together, re-differentiated and produced a new plant. Plant tissue derived from root, stem, leaf, vegetative bud, floral bud, embryo can exhibit cellular totipotency, possess the cellular totipotency. So, totipotency, application of totipotency. Totipotency character of plant cell has been used for tissue culture as follows. That is, it helps rapid multiplication of a single desirable plant, rapid multiplication of single desirable plants, multiplication for rare plants, quick propagation, and produce virus-free, disease-resistant plant, help in protoplast fusion for somatic hybridization, produce high-yielding varieties of crop plants, help in embryo rescue, or to pass the seed dormancy and develop the haploid plant. Develop haploid plant. So, in this, these are the applications for totipotency of plant tissue culture. Then various types of plant tissue culture. Plant tissue culture include cell culture, protoplast culture, organ culture, meristem culture. The organ culture includes plant organ which is separate identifiers such as anther culture, ovule, embryo, and bud. The plant part which is regenerate in tissue culture called explant. It may be a cell or it may be a protoplast, tissue or an organ. A mass of unorganized regenerated cells in culture medium is called callus. And suspension-free cells of callus in a liquid medium is known as suspension culture. The regeneration capacity of ability of plant to develop in the whole plant is known as a totipotency, which reveals each cell capable of giving rise to a complete plant. The entire vegetative produced descendants of somatic cells are collectively called clone. The entire vegetative produced an individual member of a clone is called ramete. So, in this way, totipotency is another method for plant tissue culture and hybridization of plants. So, practical applications of tissue culture. practical applications of tissue culture, that is rapid clonal propagation, soma-clonal variation, transgenic plant, induction and selection mutations, resistance to weedicides or insecticides. Then meristem culture is important in tissue culture. One can use an explant that contains pre-existing shoot meristem and produce shoots from such. Such cultures are known as meristem culture. The explants commonly used in meristem culture are shoot tips and nodal segments. Meristem culture is carried out in potato, banana, orchids, sugar, strawberry, and others. The explants are cultured in a medium containing cytokinin. The plantlets of obtained are subjected to hardening and ultimately established in the field. And it is culture for production of various virus-free plants, germplasm conservation, production of transgenic plants, and rapid clonal multiplication. So, with this, we have to finish that plant tissue. Now come to another important topic, that is micropropagation. Micropropagation. Tissue culture training or technique is useful in rapid and mass multiplication of plant material is called micropropagation. Is used for this purpose. Micropropagation refers to regeneration plants from isolated meristematic cells or tissue from somatic cells. It is also known as microcloning. Micropropagation can be used for rapid multiplication of crop plants, which are difficult to propagate sexually, or those vegetative propagate species in which the rate of multiplication is slow. Micropropagation can also be used for mass multiplication, superior hybrids, and so on. Micropropagation has several advantages are given below. It ensures pathogen-free. Micropropagation ensures pathogen-free, or which is called disease-free or disease-resistant, and it produces healthy status that is called propagules. Propagules are small plants developed by micropropagation. So propagules are small plants developed by micropropagation. It helps in rapid multiplication of a material. So micropropagation helps in rapid multiplication. A large number of propagules can be obtained from a single plant by this method. The material multiplied by this method can be maintained in a small place. Plants can be maintained in vitro in a pathogen-free state. In dioecious species, that is separate male and female plants, plants of one sex may be more desirable than those of the other. That male asparagus or a female papaya plant. In such cases, plants of the desired sex can be selectively multiplied by micropropagation. In case of forest trees, mature elite trees can be identified and rapidly cloned by this method. In case of forest trees, mature elite trees can be identified and cloned. So micropropagation can be carried out throughout the year. Micropropagation yields results faster than the conventional breeding. It is mostly used in horticulture, floriculture, and forestry. The technique has been used to obtain early and disease-free life in strawberries, bananas, citrus, and some timber trees. In field crops, micropropagation has been used potatoes, sugarcane. The main problem this technology is somaclonal variation or variation in chromosome number due to continuous subculturing. Then applications of your plant tissue culture. The use of plant cells to generate useful products or service constitute the plant biotechnology. In plant biotechnology, the useful product is a plantlet. The plantlets are used for various purposes. All the cells in callus or suspension culture are derived from a single explant by mitotic division. Genetic variation present among the plant cells of culture is called somaclonal variation. A gene that transferred into an organism by genetic engineering is known as transgene. An organism that contains and expresses transgene is called transgenic organism. A transgene can be introduced into individual plant cells. The plantlets can be regenerated from these cells. These plantlets give rise to highly valuable transgenic plants. Mutagens are added to single-cell liquid culture for induction of mutations. Tolerance of stress like pollutants, toxins, salts, dirt, flooding, etc. can also be obtained by providing them in culture medium. Woodicides are added to culture initially, and the resistant cells are then regenerated from plantlet and plants. Both apical and axillary meristems are free from the various virus, and embryos which normally do not survive inside the seeds can be grown in tissue culture to form new plants. It is useful in interspecific hybridization. The next important short course since is your single cell protein. Then you come another important biofortification. Single cell proteins, the source mixed protein extract from a pure and mixed culture of bacteria or algae or filamentous algae fungi yeast is used to substitute for protein-rich foods in human and animal feeds. That is called single cell protein. Or again, the sources of mixed protein extracted from a pure or mixed culture of bacterium, algae, or filamentous fungi or yeast. That is called single cell proteins. A protein extracted from mixed culture bacterium, algae, filamentous fungi, yeast, that is called your single cell proteins, and it is a substitute for protein requirement for foods in humans and animal foods. Nutritional composition of recipe. All are good source of amino acids except few which contain low amount of methionine. Methionine is amino acids. All are relatively high in nitrogen content. Also contain adequate level of hydrocarbons, carbohydrates, lipids, minerals, and they are excellent some source of vitamin B. SCP or single cell protein may provide the way of bringing the protein gap in protein hungry world. Microorganisms, algae, yeast, filamentous fungi, bacteria. From algae, that's Spirulina maxima or Chlorella species or Phenidesmos species, Spirulina, but Spirulina is the most important algae from which proteins can be derived. spirulina algae, chlorella algae, and Cynidesmos algae. From that algae, proteins are derived and that are called single cell proteins. Yeast, Saccharomyces, and Candida, Pichia species, and filamentous algae that Agaricus, Aspergillus, Penicillium, bacteria, Bacillus species, Mycobacterium, Cellulomonas bacterium, from which the single cell proteins can be obtained, which can neutralize the protein deficiency in human beings or any other animals. Then now come waste fermentation substrate. Bacteria, yeast, algae, and molds can be grown on a wide variety of fermentable material, including food waste such as cheese, potato processing, cannery, coffee waste, industrial waste such as sulfite liquors from papyri industry, combustion gases, and cellulose waste like baggage, straw, molasses, etc. Waste fermentation substrate. The conditions required for growth microorganisms differ according to different types of microorganisms. Under suitable conditions of temperature, pressure, pH, and water and substrate, the large amount of cell crop can be cultivated. The advantages of single cell proteins are microbes can be grown on material like wastewater from potato processing plants, straw molasses, animal manures, and space. It is an alternative protein source for animal, human nutrition from certain beneficial microorganisms. It provides nutrients rich in proteins, minerals, fats, carbohydrates, vitamins. It reduces environmental pollution due to high rate of biomass production and growth. Large amounts are produced. Microbes like spirulina, Methylophilus, Methylotrophus can be grown industrial scale as source of good protein. So spirulina and Methylophilus and Methylophilus and Methylotrophus bacteria, spirulina is an algae from which we have to get a good source of protein. Harmful effect of SCP, a single cell protein. If taken in excess, then recommended dose 2 gram per day. 2 gram per day, single cell protein can be taken. SCP can be harmful. The high nucleic acid content in SCP leads to stone or gout. The high content of nucleic acids content in SCP leads to stone or gout. So it's not necessary to take a more amount. Biofortification, another important short note. Biofortification is the method for developing crops with higher levels of vitamins, minerals, proteins, and healthier fats to improve public health. So developing crops with high levels of vitamins, minerals, proteins, it is the idea of a breeding crop to increase their nutritional value. This can be done either through the conventional selective breeding or through genetic engineering. Biofortification differs from ordinary fortification because it focuses on making plant food more nutritious as the plants are growing rather than having nutrients added to the foods when they are being processed. The objectives of biofortifications is to improve protein content and quality, oil content and quality, vitamin content, micronutrient content, and mineral content. So these are biofortifications and single cell proteins. These are two short notes important short notes that come from your exams. With this, we finish this chapter of the plant breeding. Next, we will start another new topic.